Hello everyone, my name is Yoav Blidai and I'm a fresh med school graduate and a research fellow at the Advanced Ophthalmic Imaging Lab at NYU Department of Ophthalmology. I'm here today to talk about our work on the microstructural deformation that occurs deep in the lamina cribrosa of healthy non-human primate eyes. Here are the author's disclosures. The lamina cribrosa, or LC, is approximately 250 microns thick meshwork of collagen fibers within the optic nerve head. As retinal ganglion cell axons exit the eye, the lamina cribrosa provides them structural and nutrient support. It is established that the lamina cribrosa is where glaucomatous nerve damage occurs, but the exact mechanism is still not well understood. It has been shown that several macrostructure parameters, such as thickness and depth of the lamina cribrosa, and microstructure parameters, such as beam and pore size, connective tissue density, are associated with glaucoma. Furthermore, the lamina cribrosa microstructure was shown to be complex and to vary axially and transversely. Nonetheless, many studies still concentrate on the anterior laminar surface, disregarding the complexity of this significant structure. Therefore, the purpose of this study is to analyze the microstructural deformations that occur in the deep lamina cribrosa under intraocular pressure, or IOP elevation, and to examine if anterior lamina cribrosa deformations are comparable to ones that take place within its depth. We hypothesize that the deformation response will be eye-specific, and that most laminas will deform unevenly throughout their depth. To test our hypothesis, we investigated non-human primates with acute IOP elevation while maintaining the normal intracranial pressure, or ICP. We cannulated the lateral ventricle of the brain and anterior chamber of the eye in healthy adult monkeys. Gravity-based perfusion was used to control IOP, while ICP was maintained at normal opening pressure. We then modulated the IOP, while OCT scans of the optic nerve head were acquired. We first set the IOP to a pressure of 15 mm mercury, marked as the end setting, and imaged the optic nerve head at this baseline pressure. We then raised the IOP to 30 mm mercury, marked as H1, and imaged the eye. This followed by a raise to H2 setting at about 40 mm mercury and imaged again. In between each setting, we waited for 5 minutes to compensate for the viscoelastic effect of the scleral canal and lamina cribrosa. OCT images were manually registered in 3D across all pressure settings using distinct microstructural elements to ensure that we were analyzing the same area of the lamina cribrosa. Using a semi-automated segmentation algorithm and ImageJ, which was previously described elsewhere, microstructural elements of the lamina cribrosa were acquired. For each eye and at each pressure setting, we acquired these measurements. Beam thickness, marked in blue here, pore diameter, marked in red. The beam to pore ratio, or BPR, was computed by dividing these two parameters. Connective tissue density, or CTD, calculated as the area occupied with beams, marked in yellow here, divided by the entire analyzed area. We also computed mean pore area, in green here, and mean pore aspect ratio. The results of 8 eyes from 7 monkeys were analyzed. Each OCT stack was registered to all others and split into individual frames, each at a 1.95 microns depth interval, for which the average microstructural parameters were calculated. These values were analyzed with depth and compared between the 3 IOP settings. For statistical comparison of the deformation magnitude differences with the depth, we averaged the values measured at the 5 anterior most unfossed frames and compared those to average values from 5 frames at a depth within the lamina that demonstrated maximal change. First, I want to explain the graphic presentation of our result using one eye. On the vertical axis, we present the beam to pore ratio, as this parameter was previously found to be associated with glaucoma. The horizontal axis follows the lamina cribrosa depth in microns, with 0 representing the most anterior analyzed frame. The blue line demonstrates the BPR response at baseline IOP, the purple line at H1, and the red at H2. We can appreciate a linear response to increased BPR that occurred with each pressure elevation and that was mainly in the anterior middle lamina cribrosa. Here we can examine the BPR response to IOP elevation in all 8 eyes. Quantitatively, we noticed that half of the eyes, the top four, showed evident BPR expressed deformation in response to IOP, while the rest did not. Among deformed eyes, three main patterns were noted. 1. An increase in BPR in H1 and H2, mostly at the anterior and middle depth region of the lamina cribrosa, with no deformation in the posterior lamina, shown here in eyes A and D. 2. An increase in BPR that is most pronounced in the anterior and posterior segment of the lamina, shown in eye B. And 3. A marked increase in BPR limited to roughly the posterior third of the lamina cribrosa, shown here in eye C. To differentiate between the two groups, the IOP deformers and non-deformers, for each eye we calculated these parameters. BPR absolute difference was calculated as the average of the delta in BPR at each frame throughout the lamina cribrosa depth. BPR difference standard deviation was the standard deviation of the BPR difference values, representing the variation in response. The result of this analysis, color-coded here from the lowest value in red to the highest in green, demonstrate the distinction between the groups in both parameters. Next, we compared the baseline features between the groups. While we did not find a difference in age or optic disc parameters, laminar cribrosa microstructure parameters showed interesting results. Connective tissue density was significantly higher in eyes that show IOP-induced deformation. BPR was also higher in deformed eyes, but this difference showed marginal potential significance, most likely as a result of our small sample size. Moreover, the mean poor aspect ratio showed a very small but significant difference between the groups. To test our second hypothesis, we compared the beam-to-pore ratio at the anterior lamina to an area within its depth that exhibited absolute maximal change under IOP elevation. The depth location of maximal change varied substantially between eyes and ranged between 25 microns to about 200 microns. The median absolute difference in BPR from the anterior surface was 16% in H1 and close to 20% at H2. All eyes had an absolute percentage of change that was significantly higher than a threshold of 10%. We chose this threshold as past studies showed that deformation of over 10% were associated with astrocyte activation and neuronal damage. To summarize, the lamina cribrosa is a three-dimensional dynamic organ that unevenly deforms when exposed to IOP elevation. While some eyes deform substantially, others might show only minimal deformation. We have shown that eyes that deform have higher baseline connective tissue density and beam-to-pore ratio and lower pore aspect ratio. When compared to its anterior surface, all eyes exhibited deformation that exceeded 10% at some point throughout their depth. Therefore, a microstructural study of merely the anterior lamina will not capture substantial deformations that occur within its depth. I would like to thank ARVO for inviting me to speak, to all my fantastic colleagues at the NYU Imaging and Neuroimaging Labs that supported this research, and our funding support at the NIH. Thank you very much.